DNA interest group, we meet at the Iowa City Public Library once a month. As I mentioned, this week's meeting is part of DNA Day, and so National DNA Day is April 25th, and so a day recognized to celebrate genomics through awareness. National DNA Day was sort of officially recognized by Congress, motivated by the finishing of the Human Genome Project, and so sequencing the Human Genome Project, completion of that in 2003. And the motivation is to bring together the public, teachers, educators, students, to celebrate genetics information. So tonight's program is part of our DNA Day activities that happen to span a week instead of just April 25th. Tomorrow, we'll talk a little bit more about this. We're going to have another event here at the Iowa City Public Library. And so we're going to have a DNA privacy checkup at 4 o'clock to 7 o'clock tomorrow afternoon. And this will be a come and go event on the second floor in the digital media lab, particularly if you want to go through your privacy settings on a DNA test or get some hands-on instruction with using DNA test results. Students in the Personal Genome Learning Center will be there to help you. Friday, there will be the biology seminar, and I'm Brian McAllister, and I'll be giving that biology seminar on Friday. And so that's our DNA Day seminar, and I'm going to talk about the expanding footprint of commercial DNA tests. This particular meeting here that we have at the library is part of those activities of there's being a lot of interest out there in commercial DNA testing, and so our monthly meetings are geared towards citizens in the Iowa City community that have an interest in commercial DNA tests and their applications. On Saturday at the Children's Museum at the Coral Ridge Mall, there's a craft activity associated with DNA Day, and so bring your kids, bring your grandkids. At the Iowa City Children's Museum, there will be, or Iowa Children's Museum in Coralville, there will be a craft day, 10 o'clock till noon on Saturday. So nothing actually on April 25th, but we've sort of expanded around April 25th to have a number of different events this week. I hope you can join us at some of these. There's a larger poster available if you want it. There's also small little quarter-sized pages with the relevant information if you'd like to take this with you. There's also a sign-up sheet if you'd like an email about these DNA interest group meetings and the topics that we're going to have. And so there's an email sign-up sheet over there. You can take one of those and fill that out and get on the email list. So as I said, Detective Matt Denlinger from the Cedar Rapids Police Department wasn't able to be with us tonight, and we just got notified of that yesterday around noon, and so I really appreciate my compatriots, and so students Emma McCullough and Andrew Wendell, who were planning to give a 10-minute introduction to Detective Denlinger and now have been tasked with entertaining you for the next hour. And so I really appreciate their efforts over the last 30 hours to prepare for tonight's presentation. I'll also contribute as well. And so Emma is going to start us off on DNA databases and crime scene investigation.
All right, so DNA is what makes us unique. It has this special ability to discriminate one individual from another, and the composition results in our personal identity. So there is 3 billion base pairs about that you inherit from each parent, and this results in an extraordinary shuffling of this genetic information to create who you are. So this also keeps you linked to a family tree, sort of. So when we're talking about DNA in this context, we're talking about it in a forensic use. So we're talking about identifying a biological sample, maybe at a crime scene, to a suspect in an investigation. And this is kind of what we're looking at. We're looking at the methodology of genetic genealogy in this forensic context. So what we have here is each person has 46 chromosomes, 23 pairs of chromosomes that they inherit from their parents. And what this is the chromosomes are containing this genetic information that we associate with DNA. So as you can see on the right, that image there is the DNA helix. So you have the base pairs, adenine, thymine, cytosine, and guanine. And these base pairs uniquely string together to create a genetic composition that's unique to you. So this is a consequence of inheritance. So you have these strands that we're able to use in identifying suspects in forensic investigations.
Could you speak up just a little bit?
Okay. So this is what we're going to be talking about. We're talking about the methodology of genetic genealogy in forensic investigations and kind of how your unique composition of DNA and chromosomes makes up how they can identify suspects in crime scene investigations. So 2018 has been a remarkable year for using genetic genealogy in forensic investigations. So we have 13 cases here, and we're just looking at 2018. There's many more of these cases that have been solved.
It's going to move the mic closer to you. See if this mic.
That one's turned off.
Oh, you've got it.
Maybe
Okay.
move your lapel mic up just a little.
And then shout.
Turn it up, Maeve.
What? I turned it up. Oh, so that should make a difference.
Okay.
Over there.
Thank you. Okay, so as I said, genetic genealogy in 2018, it's been a remarkable year. We've seen 13 plus cases of forensic investigations involving genetic genealogy and solving these cases. So what I have up here is 13 different cases that have been solved with genetic genealogy, and what we're talking about there is using family tree and DNA combined to solve cold cases that have been, up in the air for 30 plus years, things like that. So as you can see all the way to the left, April 24th, we have the Golden State Killer. That was a big headline that kind of jump-started the year in the amount of cases that we were going to have. And we talked about in a previous DNA interest group that kind of what this looked like. And since that time, last year, a lot has happened, a lot has advanced. So 2018 has really been a jump start in using genetic genealogy in forensic science cases. And basically, also another note to make too is that anybody that pleaded innocent in these cases, these things have not gone to trial yet. So this genetic genealogy evidence has not been used in trials yet. So we're still waiting to see what that kind of looks like. So advances are still to come past the year of 2018 into 2019. So this isn't even including all of the cases. This is just kind of a snapshot of the last year and how far we've come in that amount of time.
So we've seen heavily where we are today in 2018 and what has gone on in the past year, especially with the Golden State Killer suspect being arrested nearly a year ago today. But in order to fully understand this concept, we really need to understand where we have been and where this technology really has originated from. So in 1999, there was a case in the Netherlands, right along the border of between Belgium, Germany, and the Netherlands, where they all converged. It was a brutal rape and murder of a young girl in a very small town. But right outside of this town was a refugee center. So a lot of different nationalities and ethnicities were at this refugee center, and the local police didn't necessarily know how to identify who it was, since the only evidence was a trace evidence, presumably from the suspect. So what they did was that the local government decided that it was a serious enough case that they were going to go around to the neighborhoods and specifically the refugee center and collect DNA samples. Essentially what they were doing through this process was building a DNA database full of different DNA samples from different people, or different people with different ethnicities. So just kind of the structure of that makes it sound very similar to what we're seeing today in direct-to-consumer DNA databases. Through this testing, they were able to identify relatives of the suspect, since presumably an entire family would move to a refugee camp. And so they were able to find relatives and through that narrow a suspect list down. They were eventually able to find the person who had killed the young girl, but in the process built the foundation to what we have seen today in, especially in 2018 with solving these cold cases. And so today what we're really focusing on is especially the Michelle Martinko case, which was December 1979, just in Cedar Rapids. But we'll also touch on the Joseph DeAngelo Golden State Killer case, since that was really kind of the linchpin that set off all of these cases to be solved with this method. So this is a timeline essentially laying out to where these cold cases first became cold, with the Golden State Killer case in 1974 officially going cold in the Sacramento Police Department, and then the Michelle Martinko murder in 1979. In 1984 was a really big discovery in the UK by Sir Alec Jeffreys. He discovered the method, or really founded the method of DNA fingerprinting, which was really the first way that DNA was being used in forensics. And we'll touch later, just right after the slide, but the DNA fingerprinting was then first used in 1986 to convict someone. Colin Pitchfork was the first one to be convicted on DNA forensic evidence in the UK. And then throughout time, as you can see, we have different landmark cases, different periods where commercial DNA testing really took off in the late 1990s and then has carried through with as latest as being Ancestry DNA kits being made available in 2012. And as you can see, there's a bit of a circle story here with the Golden State Killer case being solved and then the Michelle Martinko case seeing an arrest nearly 30 years to the day. So this would be the DNA fingerprinting and what it looks like. On the left there, or on the right there, is going to be Colin Pitchfork. He's the first person to be convicted on this evidence. But this is essentially what a DNA fingerprint looks like. It's splitting up DNA into different segments based on its length and then separated out. So how you can interpret this would be if you had 4 different DNA samples, like that in the figure, the right of the figure, you can match it with what you see in the crime scene. And so then the different segments and the different lengths, and if they match, presumably that is the match that matches your sample from the crime scene. This built the foundation for DNA forensics in the police department. And since we've seen incredible advancements in forensics, especially with using DNA, and this was really first seen in the CODIS system, which has been made available locally, statewide, and federally into labs in order to help solve cases. But with advancements, there's always limitations. And Dr. McAllister will talk to you about that.
So as Andrew introduced, DNA fingerprinting was an early use of DNA as a forensic tool, using this uniqueness of each of our DNA profiles and developing a method in order to assess one individual to another to discriminate among them. The CODIS system was a standardized methodology that was developed and adopted by different law enforcement agencies. And so federally, at statewide level, locally, there was a standardization of the DNA platform in terms of how a particular individual that's found guilty, that sample would go into the database and the type of data that was obtained from that individual. And so this represents an earlier version of the CODIS system that's based on these 13 core positions or loci distributed throughout the human genome. And so as Emma indicated, we have 23 pairs of chromosomes. This is a diagram that illustrates those 23 pairs of chromosomes. Each of these little boxes is one particular marker or locus that's used within the CODIS system. This is a representation of the standard 13 marker test that's now been upgraded to a 20 marker test as the standard that's used in forensic DNA analysis. And there's also overlap between the system that's used by US law enforcement agencies and also European law enforcement agencies. And these particular markers are built on a specific type of change within DNA sequences that I'll illustrate in just a second, but that's this STR locus, which stands for the short tandem repeat, and I'll show you what that short tandem repeat looks like. And the whole idea of this system is by looking at an individual's profile in terms of their series of variants that they have at each of these 13 or 20 short tandem repeat markers, that's a unique signature of that individual that can be used to identify a crime scene sample with the personal identity of the individual that's responsible for that sample being at the crime scene. And so here's an illustration of what that short tandem repeat marker looks like. And so in this case, I'm illustrating a sequence of DNA, and these are short sequences that are repeated in tandem. And so in this case, we have this CA repeat, whereas this particular variant has 5 copies of that CA repeat. This particular copy of that variant has 4 copies, 3 copies, 2 copies, or 1. And so that's the short tandem repeat type of variation that exists that produces different lengths in terms of this region within the DNA sequence. And so a marker is developed to simply assess what is the length of that region, and we know that length variation exists within the human population by the number of repeats at those individual marker loci. And so this is short tandem repeat standing for this short repeat that's present in tandem, and however many numbers of repeats, that gives you a particular length that's present on an individual chromosome. These are easily assessed as illustrated here, and so this is a diagram that's essentially capturing a genetic profile at a series of short tandem repeat markers. And so this would be one particular marker, and in this case, these are 2 then linked variants that represent the 2 chromosomes of that individual. One they inherited from their mother, one they inherited from their father, and in this case, we get a measure of the length of those 2 variants at that marker. Here's another marker, and there are 2 linked variants at that marker, 2 linked variants at that marker, and so a number of different markers can be ran in one particular assay, and so these are really easy and quick to obtain from a DNA sample by running a series of markers sequentially in one assay. Here's another assay with a few markers in it, and another assay with another few markers. So you can build up quickly those profiles at the 13 or 20 loci that are being assessed in a particular DNA sample. So here's just an illustration of a simplistic view of using this type of technology. And so in this case, I'm illustrating 4 different loci. So CODIS system is either 13 or 20. I'm illustrating a case of 4 different loci, and in this case, I have at each locus 5 different repeat lengths, and so we have 5 different variants or alleles that are present at each of those, corresponding to sort of these 5 different length differences. The reality is for human STR loci, there's generally more than 5 different length variants, and so you have more than 5 different alleles that are present at a particular locus. And so a profile of an individual at these 4 different loci may look something like this. And so a combination at the D locus of the length 1 and length 3 variants. At the C locus, we happen to have 2 of the same, so 3 length of 3 variants. Here's a 2 and a 4, here's a 1 and a 2, and so that's a particular profile at those markers. And simply based on the simplistic case, where we have 5 different variants at each of these 4 loci, there would be about 50,000 different combinations of variants that you could have for different genetic profiles. And by using the human system of 13 and even 20, then you're essentially exceeding the human population whenever you consider the number of combinations you can get across those different markers as you're assessing them in terms of combining those particular variants together to make a unique genetic profile. And so this is a very powerful system for identifying uniquely individuals and also excluding individuals that do not match a particular profile as well. And you can build probability models based on the prevalence of these particular variants within a population and use that to predict how common you would expect a particular genetic profile to be at that marker and that marker and that marker and so on and so forth. That's the case for an individual out of the human population, considering how these can be combined more generally. Reality is that related individuals are going to share variants. And so related individuals, such as illustrated here, if you have a DNA profile and a full sibling, those particular individuals share a mother and a father. And so in this case, you would also expect them to share some of their variants because they would inherit them from their mother and father. And for full siblings, you would expect them to share about 50% of those variants would be identical. And so you might see a situation as illustrated here, where in this case, they both inherited this 3 variant from the same parent. And then in this case, they inherited different variants from the other parents, or the other parent. In this case, they happen to inherit nothing in common. And so this individual got 3 and 3 from the 2 parents. This individual got then the 1 and 4 variant from those parents. In this case, they happen to be identical. And so they got the same thing from mom or dad. They got the same other thing from mom or dad. And so in this case, they are identical in that instance. In this case, they share. In this case, they both inherited the 1 variant from mom or dad. And then the other parent, then they got 2 and 5, which would be the other chromosomes of mom and dad. So this type of sharing of not complete profiles has been used in forensics in terms of familial searches on the CODIS database. And so when you identify individuals that aren't a complete match to a DNA profile in the CODIS database, yet they do share some variants in common, that can lead you into a situation where you may start looking for, if there's a full SIB or a half SIB that may be your suspect in the crime, the sibling being in the CODIS database, and then the suspect then being related to that individual. However, this system, because it's only based on 13 or 20 loci, breaks down if you look at more distant relatives. And so if you look at first cousins, considering the 13 locus case, you would only expect to see about 3 or 4 variants in common whenever you're considering a first cousin related to someone that has their profile within the database. And so Familial searching isn't very powerful using the CODIS system because it's not based on that many loci, but it is very powerful for excluding individuals and identifying matches of high probability whenever you have an exact match. And so this is an illustration of STR testing. And so those STR loci, independent of CODIS, there has been expansion of this commercial DNA market that's based on these single nucleotide variants. And so single nucleotide polymorphisms are single nucleotide variants that occur within DNA sequence. And this is the technology upon which Ancestry DNA is built and 23andMe is built. They're based on assessing these single nucleotide variants, but assessing many of those. And this represents the status of sort of the position of one prominent genetic genealogist in February of 2015 in regards to the use of the genealogical platform of this SNP testing in relation to law enforcement. And so the legal genealogist from her blog, this essay published in 2015. So when a cousin asks you, once again, whether his genetic genealogy test can be used by the police, remind them, once again, that the police don't particularly want it. And they don't need it. First, the tests we take for genealogy aren't all that useful to the police. Our tests tell us how we are like other people, our family members who share common ancestors with us. The CODIS markers focus on parts of the DNA that make us unlike other people and set us apart as individuals. And so as Emma sort of introduced, as 2018, this has changed. And so it's now pretty clear that law enforcement is interested in commercial DNA platforms and is interested in the SNP-based inference of relationships and is interested in using that for identifying suspects in criminal cases, as we'll illustrate. And so just a little bit more detail on the SNP platform. And so single nucleotide polymorphism, or SNP, refers to these positions in our DNA where we are variable. There is this chip that can assess many different sites that are known to be variable in the human genome, and the common testing platform used by Ancestry and 23 and Family Tree DNA is based on assessing about 600,000 of those particular sites spread throughout our genome. And so with that, you can make really strong inferences in terms of relationships. And so many of you are likely done these tests yourself and use this as part of your genealogical research because these are very powerful in terms of identifying relatives in the database based on that technology. And because of that use, that has really powered the growth of the market. And so this is a graphic that illustrates the growth of the Ancestry DNA database, the growth of the 23andMe database, the other databases, and this has really grown quite rapidly over the last several years in terms of the number of participants that have taken advantage of these tests, and to a large extent are interested in using them to find relatives and using those relatives that they discover to inform their family histories in terms of their genealogical research. And so these are powerful because those SNPs, as you assess 600,000 of them across the genome, you can essentially sort of paint chromosomes in terms of these series of identical segments as illustrated here as you take two individuals that test in the database. And so these two individuals are testing on that SNP platform. You can identify these individuals as relatives because they share these identical stretches of chromosome that they inherited from this common grandparent in this case. Or they have identical stretches of chromosome here that that in this case the red's not there, but the blue's there, and so we have that blue bit that's identical, and so there's that region of identity, and in this case we have that purple bit, so that region of identity there, and so we have stretches of the chromosome that are painted by the unique series of SNPs that are identified within this person's sample and within that person's sample that trace back to these common ancestors, these common grandparents. And so this is very powerful in terms of matching you with relatives because you share common ancestors with those individuals, and first, second, third cousins can be identified within the database. The power of the technology sort of diminishes beyond that point. And so GEDmatch, which has come to the forefront of the use of the SNP test in forensic genealogy, GEDmatch is this third-party site where you can take your DNA profile that you've obtained from Ancestry, you can download that raw data and upload it to this site, and GEDmatch is a platform that you can use to work with your DNA data with a lot of different tools that are helpful to individuals that want to investigate their relationship to other people within the GEDmatch database. Also accepts 23andMe data or other commercial data, and so this is a case where the hobbyist has developed this, and the hobbyist uses it in terms of a platform for working with genetic data. This was also open then for law enforcement to use to put their samples into this database and use the individual profiles that are available within the GEDmatch database to then identify suspects that in particular criminal cases, such as the Martinko case, such as the Golden State Killer case, identify those suspects based on the matching between those suspects and other people that are in the database. And I'll go through in some detail sort of what that looks like using GEDmatch as this tool for doing matching. And so GEDmatch, it's a publicly available website, and we did have last June at our DNA interest group a program on the use of DNA data in forensic analysis. We had Anya Prince, a professor from the UI Law School, here as part of that program, and partly what she communicated in that particular program is putting your data in GEDmatch is similar to discarding a straw and police having then access to the straw that has your DNA in it that's discarded in a trash can, and they can then retrieve that straw and get the DNA from that. Uploading your data to GEDmatch in legal terms is similar to that activity of discarding a straw. Yes?
Brian, how big is GEDmatch?
It's about 1 million people. About 1 million samples in there. So it's free to upload your data. It's available for anyone to upload their data. You download your data from the company that did the testing, and then you upload your data to this particular site. And there's a lot of tools that you can use to work with your data. I'm going to show this one-to-many matches tool, and I'm also going to show a tool of using this matrix of comparison as well, but there's a variety of different tools available to the genealogist that wants to use this, and also, in this case, as we've seen in the past year, for law enforcement to use it as well. So, you can really work with any kit that's available in GEDmatch. Kits have been uploaded. You essentially have access as long as you can either find it in a match list or know the kit number, and so you can work with those kits. And this shows the one to matching the database. You're getting a list of matches to this particular sample within the GEDmatch database. And so we take this sample, we get a list of matches, and in this case we get a measure of how similar the profile is. And so this would be law enforcement putting this profile in, and we get a series of matches to that particular profile, in this case indicating that connection about 2 to 3 generations out up to 4 generations. In this case, we have 2 individuals that have links to their genealogy information. And so Wiki is a public resource for genealogical information. And so all we need to do is click on those Wiki links, and we can see, wow, we have genealogical information now for 2 of our matches to our crime scene sample, and it's publicly available to us. And so one of those happens to be me. And so in one case, in my case, I get these as my ancestors, and I simply look through my 254 ancestors as linked that appear on that link, and I take the other individual and look at the ancestors on her link, and boom, I see Martin Brighton, Mary Long, Martin Brighton, Mary Long. There's a common connection between those 2 individuals. It's potential that that particular connection there is a common connection with my sample, that I'm wanting to know who the identity of that sample is. And so in this case, I can go to WikiTree or other sources of genealogical information, and those 2 individuals that have this common ancestor of John Bryant and his wife Mary. And so here's their relationship to that couple as obtained from WikiTree. And so here are our 2 matches here. Here's our crime scene sample. Here's the metric of how similar our crime scene sample is to me. Our crime scene sample is to this. And so in this case, here's me, M351. Here's this case, A497. This is a case of comparing me and A497, and then if you do a little bit of work, you figure out that A63 is also my father, and so there's the green case of my father. Our crime scene sample here shows a similar relationship to both of these individuals, so 90 to here, 106 to there, and this is an indication of relationship. And that particular metric of how similar they are is an indication of about a 2nd to 3rd cousin relationship. These individuals are 3rd cousins, so sharing these common ancestors, so historically they're 3rd cousins, and this would suggest that this particular individual may be a descendant of another sibling of these individuals here, so another child of that couple, that particular suspect may be a descendant of them. So what you would want to do as an investigator is to know if this couple had any other children. And so investigate essentially their descendants to see if you can find a line and that leads to a person that's in the right time, the right place, that person's the right age, that person's the right sex. And so a personal identity that ultimately fits with the crime of interest. And so that's the essence of using GEDmatch to then go out and sleuth and identify a potential suspect by now wanting to do some genealogy and figuring out who else is descendants of that couple. And Andrew will talk about this in a bit more detail in terms of the Martinko case and what's been reported. So GEDmatch is now upgraded to this GEDmatch Genesis, and so this is the Genesis version, so it's just changed slightly, but it's essentially the same thing as I've shown previously. The old one sort of is not being supported anymore. This is the newer version of GEDmatch. So.
Martinko case compared to what we just went through hypothetically. And just reviewing kind of the facts to what's been reported through the Cedar Rapids Gazette about the Martinko case. Michelle Martinko was found December 20th, 1979 in the Westdale Mall parking lot. It was a brutal murder with very little evidence in regards to who the suspect was. A DNA profile was created based off of a blood sample on the car gear shift. And so back in 1979, this DNA profile was created, was stored, but once the case went cold, The Cedar Rapids Police Department was continuing to work on it throughout time, but really wasn't touching the DNA sample just because that DNA technology really wasn't there yet and really wasn't established, and especially this method was not established. So in 2006, another DNA profile was created, and it was uploaded to CODIS. The match came back negative, so as we went through before, there was no person in CODIS that was closely enough related in order to get a hit. In 2016, Parabon Nanolabs began being contracted through Cedar Rapids Police Department in order to use their genetic testing profile. And we'll go through Parabon Nanolabs in a second, but basically what they did is they created a snapshot and a phenotypic expression and sketch that you may have seen. It was made incredibly public. It was the source of hundreds of leads, or hundreds of tips in 2016. And then in 2018, a genealogical database, specifically GEDmatch through Parabon, was used to create the suspect list, which eventually led to the arrest of Jerry Burns. So this is what Parabon looks like. Parabon is specifically a website, a company that works for law enforcement agencies in order to help solve cold cases and crimes. They offer these services. So at first, like I said, the Cedar Rapids Police Department began using DNA phenotyping, that service, in order to composite a sketch of what the suspect may have looked like back in 1979, and also what the suspect would have looked like at the time of the release of these pictures. So they were guessing that approximately the suspect would be around 50 years old in 2016. But as you can see, Parabon offers two other services, genetic genealogy and then kinship inference. And it will be the genetic genealogy service that will eventually lead to the Jerry Burns arrest. So in 2016, these were the sketches that the Cedar Rapids Police Department released to the public. The top two are what the suspect would have looked like in 1979, according to the DNA profile that the Cedar Rapids Police Department gave to Parabon, and then through that DNA profile, they were able to examine the genes and create a sketch based on what the phenotype would be, similar to what 23andMe does with predicting traits, but instead of rendering a sketch, like Parabon, they just give you what you're most likely to be: blue eyes, brown eyes, etc. So they created a sketch with the suspect having short hair and long hair. And then this is a picture of what the suspect would have looked like at age 50. And then this is actually a picture of Jerry Burns at age 25. So if you compare this one to the one snapshot of the suspect in short hair, it is fairly close. But as you might be able to guess, that this method only produces tips and doesn't really produce concrete answers until further investigation. It was really the genetic genealogy which took this investigation to a different level. And so what the Cedar Rapids, what Parabon did once the Cedar Rapids Police Department allowed them to, was they tested the DNA profile compared to GEDmatch. And in GEDmatch, they found the orange box is going to represent what the crime scene sample was. And this green circle is going to represent the hit that they got, which was approximately a second cousin. So they knew and were able to assume that the person that they got and then the crime scene sample were going to be related through the great-grandparents. And like Dr. McAllister just went through, how that would be the convergence point of these two trees. So the next step for the Cedar Rapids Police Department would ideally have been to go through the 4 sets of great-grandparents that the green circle shared, or had, and tested living descendants of these lines, of each of the 4 lines. And what they found was that their first 2 trials, first 2 tests of living descendants came back with no results. So they knew that it could not have been a maternal line related, that was the relation between the second cousin relationship of the crime scene and the hit they got through GEDmatch. So therefore it had to be through a paternal line. Whether or not this line was through this line or this line, it's completely arbitrary. Those facts were not made public to us. So when we created these trees, we just chose this line for the ease of it. And so they went through the same process of looking for living descendants of these lines. And what they found was that they got a hit in this line, which proved to be a first cousin relationship between the new hit and then the crime scene sample. So now they have it narrowed down to not only a great-grandparents, but presumably an uncle and aunt eventually. So what the next step would have been was that to take this first cousin hit and to essentially compile all of the first cousins that that this person has. And then presumably in that would be the suspect. Ideally, or what happened, presumably, was that the crime scene suspect had 3 brothers, the Jerry Burns and 2 other brothers. And they would all have the same relationship between themselves and the first cousin. So what the Cedar Rapids Police Department did was that they collected DNA samples from all 3 of the brothers, and the sample that Jerry Burns was caught on was a straw from a restaurant that the police department waited for him to be done at the restaurant and for the waitresses to collect, and that is where they got their DNA from. And so through this is essentially how the genetic genealogy relationship is what drew onto the arrest, but specifically it was through Parabon Nanolabs and GEDmatch, which Emma will talk about now.
Okay, so we've talked a lot about GEDmatch and how that was used to come to the arrest of Jerry Burns in the Michelle Martinko case, but now how does that relate to direct-to-consumer tests now, like Ancestry, 23andMe, those types of tests? How does GEDmatch relate to those? It's not exactly the same thing as Ancestry and 23andMe, so basically the access, and we're talking about especially law enforcement access, is different with each of these different databases. So the third-party websites are where you can upload raw DNA data from Ancestry and 23andMe that they've come up with in their own labs, and you can submit that to a third-party website and upload it on there to come up with more analysis of that DNA. So examples are GEDmatch, you have GEDmatch Genesis, which Dr. McAllister went through, and you have DNA Land. So these are very separate from Ancestry and 23andMe in terms of they do not process your DNA samples. They just take the raw data from Ancestry and 23andMe, and they process that to come up with new analysis, and they come up with their own database of about 1 million users in terms of talking about GEDmatch. Commercial DNA tests are what we're talking about when we talk about 23andMe and Ancestry DNA. So these places you actually spit into the tube and you send it in, and then they do their own DNA analysis and they send you the results. So this is different from GEDmatch in terms of that. And then you have the hybrid sites like Family Finder at Family Tree DNA and MyHeritage DNA that in terms of combining third-party websites and commercial DNA tests, so they will take saliva and cheek swab samples and they'll process their DNA, but they also will allow raw uploads from other sites as well. So it's kind of a combination of third-party websites and commercial DNA tests. So that's kind of how they differ. in terms of how does this relate to you, many of you guys might have Ancestry accounts, 23andMe accounts, and you might want to know how does the access differ between law enforcement having access to GEDmatch versus Ancestry versus 23andMe. So as you can see with GEDmatch over here, I have pulled up all of the kind of the legal or regulatory processes between their privacy policies and how people can access these databases. So you can see a major difference between GEDmatch and Ancestry and 23andMe because GEDmatch, as it talks about in the raw DNA analysis over here, the DNA can be DNA obtained or authorized by law enforcement can be submitted to GEDmatch and can be compared to other matches in the system. There is no GEDmatch does not prohibit the law enforcement to submit their samples. So the law enforcement has free access to this. It's a public database. They don't have to go through any sort of restrictions at all with GEDmatch. This differs from Ancestry and 23andMe because they are not required to release consumer information unless there is a valid legal process. So we're talking about subpoena, we're talking about very official court orders that require them to do so. So the information is safer on Ancestry and 23andMe than on GEDmatch. Ancestry talks about you have to go through subpoena, you have warrants, and the same with 23andMe where they try to resist requests from law enforcement unless they are forced to. And in both of these cases, if they are forced to, then you get an email from them saying, hello, like we're talking to a law enforcement, either state, federal, etc., and they're looking at wanting access to your personal information. And so you will get a notification about that if that ever happens to be the case. And so you will be able to mount your own legal response to that in terms of them letting you know that they want access to your information. So they will give you kind of a notification that they're using your personal information, whereas GEDmatch, public database, does not send out that notification. Law enforcement can just get on there and find matches and use that to solve forensic investigations. And so we looked further into Ancestry DNA and kind of what valid law enforcement requests were put in their specific system. So they received 10 valid law enforcement requests in the year of 2018. Mind you, they have 14 plus million users, so only having 10 valid law enforcement requests is a very small number in comparison to how big the database is, and only 7 of those 10 requests were actually answered by Ancestry DNA. They only had to provide information in 7 of those 10 cases. So they refused a lot of inquiries from law enforcement, and they basically were able to stop a lot of those requests, so user information was protected. And so this kind of goes into more of that, about kind of what jurisdiction they're looking at. So 2 of the requests were actual U.S. federal law enforcement investigations, and 8 of those were U.S. state. And then it kind of talks about the types of requests that they're looking at in terms of criminal subpoena. There was 8, civil subpoena there was 1, and then search warrant there was 1. So, and both of those, there was no emergency request and there was no administrative subpoena. So just kind of looking at what types of requests were submitted to Ancestry, and there was no, obviously there was only 10, so once again, not a very large number of those, but just looking at the statistics of how this is a very rare occurrence if it does get through the Ancestry DNA database at all. And so this kind of brings us to recent news. You might have heard about Family Tree DNA and how they're changing their privacy policies because law enforcement can have access to Family Tree DNA more so than Ancestry and 23andMe. So what we found in the news was that the FBI had already been accessing Family Tree DNA's database before sending out a notification to consumers that they were using this information. So basically how this story goes is that the FBI had been submitting these files to Family Tree DNA's database, and these files were unreadable. The Family Tree DNA genetic company was looking at this and was like, what are these files? We can't read these with our website. So they emailed this user and they were like, what are these files? Like, what are you doing? And it was the FBI, and they were looking for information that users had submitted on Family Tree DNA to solve criminal investigations. So not only had the FBI been using this database without registering DNA samples because they were submitting their own files from Parabon Labs, which Andrew talked about. So they were doing this without kind of notifying the consumers that they were doing so. So the consumers didn't know that their DNA was being subjected to law enforcement and their criminal investigations. So this is kind of where Family Tree DNA came under fire, because they sent out a email to its consumers saying, we have not been communicating with you, and we have been using your, the law enforcement has had access to your information, and basically they were changing their privacy policy while people were already having accounts on their website. So that's kind of where they came under fire, and that's how Family Tree DNA differs from Ancestry and 23andMe, is because law enforcement was able to bypass them by submitting their own files into the system and being able to compare their matches and their DNA samples with ones that were in this system already. So, and this is kind of what Family Tree DNA's law enforcement guide looks like. This is kind of how they changed their privacy policy, and that's something that consumers with Family Tree DNA should be aware of, is that it's not the same as Ancestry and 23andMe, because law enforcement now, with permission from Family Tree DNA, can go through and get consumers' DNA and match them to crime samples. So that's kind of what we're looking at here, is that they can have access to this, and how it's different, even though it's not a little bit different from GEDmatch, because GEDmatch doesn't have to give consumers notification. Family Tree DNA has more accessibility for law enforcement, and they use that permission to solve criminal investigations. So that's kind of what we're looking at with that.
Let's go ahead and do so talk about the thing tomorrow, and then we'll go to this and then have questions.
Okay, so this kind of brings us in, how does this relate to you? How does this relate to your accounts with Ancestry, 23andMe, etc.? So we're talking about, tomorrow we have basically a DNA privacy checkup. If you guys have Ancestry, if you have 23andMe, Family Tree DNA, any of those sort of direct-to-consumer tests, we go through tomorrow the privacy settings and kind of managing those, because with Family Tree DNA, especially if you have that account, in the United States, they have it so that you're automatically opted in to have the federal law enforcement to have access to it. So if you do have that account, you have to go in and you have to opt out of that if you don't want to participate in possibly solving criminal investigations. So tomorrow we're having a privacy checkup for these different databases, and that's from 4 to 7 p.m. here at the public library. So feel free to come to that if you have any privacy concerns, if you want to learn more about how to manage those settings, and that's something that a service that we provide, that the DNA interest group does.
I'll just add to that is, beyond Ancestry and 23 not being involved in law enforcement, there are potential privacy concerns you might consider in the context of those platforms as well. They recently have both introduced this map feature, putting your matches on the map. And so if you want us to look at if you appear on the map and where you appear on the map of your matches, we can go through and look at that and let you know where you stand in terms of other privacy concerns individuals may have with the various platforms. So with that, since we're sort of at the end of the hour, we do have a short clip. There's a 5-minute clip that I'd like to use as sort of a springboard into any questions you might have. But if you don't want to stay for that 5-minute clip from 60 minutes, you can go ahead and leave. But I'll take this opportunity now to thank our presenters. And then what I would like to do is to show this little clip from 60 Minutes. They had a program on solving cold cases with genetic genealogy, and this is a follow-up discussion on the 60 Minutes overtime. And so I'd like to show this 5-minute clip and then take questions that you might have. So if you need to leave, you can sort of get up and leave during this time. Restart it because we need a volume check. Let's go to the presenter. I think that was a very informative conversation after the 60 Minutes did this piece on the Golden State Killer case and this technology. So with that, I'll be happy to take any questions that anybody might have. I think we're off mic now, so it actually doesn't matter. She killed our tape because of the 60 Minutes thing. Yeah.
No, use it.
Use the mic?
Q&A part.
Okay, use the mic. She's going to keep the Q&A part. So. She'll just delete, apparently, the YouTube video that we just showed. Anybody have any questions? There's one in the back, Maeve.
Hello? Is it on? It's not on. It is on.
Never tell.
I understand how for GEDmatch the matching works, but if you have a DNA sample and you want to use Ancestry.com, are you basically getting a subpoena for the entire database? Because it would seem to me if you could limit it to an individual person, you might not need that. So I'm not quite sure how that works.
My understanding of Ancestry is they will not allow a subpoena for, say, law enforcement to put a profile into their database. And so they would not allow that. That's my reading of their privacy policy. It would only be a subpoena on information that they have for some reason. And how about you, Emma?
That's my understanding of it as well.
So what are they asking for, though? If they can't run a match and they don't know who to match it to, what information would they be getting from that?
It's kind of like how Andrew talked about the fingerprinting. It's kind of like how they match it up in the bands and stuff. So they're taking a biological sample from a crime scene and they're trying to match it to an individual and their personal identity.
But I think what she's interested in is what law enforcement is looking for whenever they subpoena Ancestry. And I suspect, and actually there's no public sort of knowledge of what the subpoenas are that have been delivered to Ancestry for access, but I would suspect it's a case of participants already being in there and maybe the finding of a half-sib or some kind of sort of recent biological relationship becomes a legal issue, and they're wanting access because of that. That would be my only guess, but really the specific information of what those subpoenas are on the Ancestry DNA database or Ancestry.com database are not public knowledge. Unless anybody else knows. I mean,
I just wondered if you could speak to the issues around the case in Germany where multiple people were tested and sort of the legalities and the civil rights issues around that and how those were obtained and just, I don't know, it seems to me that there's concerns about testing, large swaths of a population, particularly when you're talking about refugees. And I was just wondering if anybody could speak to some of those issues.
Andrew, you want to deal with issues of access to profiles differently in Europe versus the United States?
Mm-hmm. So Europe is, or I'll phrase it that way. The U.S. Values privacy and their privacy rights as individuals a lot heavier than European countries do. And in that case specifically, the European governments don't allow that large-scale DNA collection really unless it's a serious crime. And as it's defined, serious crime in Europe is defined as a life sentence or a capital offense. So if that is on the table, such as the brutal rape and murder of the young girl, and the circumstances such as it being on the border of multiple countries and with the political asylum camp being so close, that's how they allowed that. In the United States, that wouldn't ever be allowed unless a Supreme Court case ruled against that, which is essentially would have to be an amendment. So
In that case, in the Netherlands, I expect would be very different now than it was then, because the European Union has just recently passed some privacy protection laws that are very stringent. And so I suspect that now things would be very different in the Netherlands than they were there back then.
There's the right to remain anonymous. Is that what you're referring to?
It's a GDPR, the whole privacy issue with internet, genetics, I mean, all kinds of privacy issues under this new European law.
So in that case, were the samples compelled or was this voluntary?
So those were voluntary. And the idea was to look for relatives. The funny thing about the case is the guy that got caught thought that if he participated, he would get away with it. So he actually volunteered. So that's why the method of it, so the method of it, that's why it built that method, but it provided a direct sample.
Well, and as an extreme example, on the other end of the spectrum is, I think it's Kuwait that recently passed a law to get everyone's genetic profile in the entire country. And so that's the other extreme. And so looking across different legal systems, sort of their access to your genetic data varies quite a bit.
Well, but there have been police departments in the United States that have taken swabs without really the permission to do, or no, they've people that haven't been arrested, they've just been questioned and taking swabs from them without due cause.
Well, getting into local CODIS databases, that varies across different jurisdictions in the United States. And so in some jurisdictions, you can get in, I think it's county or state or some level of local CODIS database simply by being arrested for a crime. In others,
But
you
not
have to
being
be
convicted.
convicted.
That's
And so that varies in different legal jurisdictions. I'm not an expert in that, so.
Wait. No, it doesn't work for the people at home, so. Yeah, I'm talking to it.
We'd have to ask our legal expert, but I don't think the discarded DNA has ever made it to the Supreme Court. I think it's on its way. That may not last.
No, I think that the DNA from the straw has made it to the Supreme Court.
I'll look on that. For some reason, I think it has not.
As I recall from last June, I think I'd have to relook at that video. It actually is available on the library's channel. And so Anya's presentation from last June is available on the website.
Wait, I was just going to say that the library doesn't have a channel anymore, but all of the videos are available on YouTube through the library channel. So just.
They're also all linked to our Facebook events, and so every series of meetings we've had, they're linked to those if we have a video.
It's a little bit close to yourself, it does work. It's on, believe me.
My question is a little bit different. What about this DNA that shows up in a crime scene that isn't the right person? Like this guy in Iowa City with the blanket. And there was another case in California where luckily he was in jail, so he did his DNA didn't. So that's the one thing that kind of prevents you from asking all your friends to get their DNA done because it's a good idea.
So, I mean, that's part of the consideration whenever you are participating and decide to put your data in GEDmatch is, okay, so if you have a series of cases or if you have good evidence that sample comes from the crime scene, because part of it is matching the crime scene sample with a personal profile to develop a suspect, but what's the validity of that crime scene sample?
Because they went to the same McDonald's and used the same restaurant.
And so the specific case that you referenced that Iowa City is currently being sued for is an instance where it's being challenged in terms of how that sample led to a particular match for a rape that happened within an individual's apartment. That individual then was prosecuted as a participant in the rape, in addition to another suspect that was found guilty in the rape. This particular person that was also prosecuted because his DNA profile matched a sample from the victim of the rape. He was found not guilty in the trial. The rape happened in his apartment, and essentially the defense argued. And he's saying he's in Chicago, so that all of his defense is I was in Chicago. The defense also challenged whether or not, simply because the rape happened in his apartment, that his DNA could have wound up as a match, and the jury sided with that argument, and he was found not guilty, and now he's turned around and has sued the city of Iowa City, Johnson County.
It does kind of show you the danger of it.
So that's the issue of the crime scene sample and how much evidence there is in terms of that being from the suspect, and that particular person, once you identify who that person is there other corroborating evidence to show that that person committed the crime? And so that would be sort of the legal set of arguments that are going to be brought to trial to try to convict the person. You had a question?
Does HIPAA offer any protection?
No. So the question is if the Health Information Privacy Act offers protection, and it doesn't, because that's all about health. There's a question up here in the front row.
All right. Just a quickie, but those like cold cases, the DNA, right? If they collect a blood sample, like how long is it good for? Does like DNA die or something? I have no idea. Kind of curious.
So DNA can be stored, and so that was the big thing with the 1979 case, is that even though trace evidence is there, the cold case can essentially stay open and be investigated this way because they can store the DNA that they would have extracted.
Yeah, and a lot of it comes with advancements in DNA technology,
Yeah.
so they can store DNA from past cases that they weren't able to solve, and then when we have new technology to be able to investigate these cases, we're able to go back to that stored DNA and actually analyze it. Mm-hmm. So that's why cold cases are being solved now.
Well, and in the case of the Golden State Killer example, there were multiple samples of DNA, and the one police department in Sacramento that sort of spearheaded it was running low or out of their sample, and so they actually went to another jurisdiction that at one point had matched and so been shown to be connected because they had essentially, whenever they collected DNA evidence, they would split it, put one in storage, and then use the other one. And so in the case of using it for the genetic genealogy, they went to the one that had never been accessed, had immediately been put in storage whenever they sampled the crime scene, and they used that one as the sample to investigate the case.
But can't they get DNA even from ancient fossils and things like that?
Yes, and so DNA is a very stable structure, and so its molecular structure is quite stable. And as long as you don't have a chemical environment that's very damaging to DNA, it's likely to be quite stable for a long period of time. So there are things that are damaging to DNA. I think there's now a service out there now that sells a solution that you can sort of use to wipe surfaces and clean it of your DNA. And sort of this modern terminology of technology, it's sort of DNA away or something like that, that you can use to sort of clean the surfaces in which you touch and remove your...
Trace DNA is everywhere.
Any other further questions?
So thank you for your attention.
Next
Come,
month, what's happening?
come back tomorrow. Talk on Friday if you're interested in commercial DNA market, sort of the spectrum of that. Next month, we're going to have a program by UD Brandenburg on finding the German hometown of your ancestors. And so this is a presentation modified that was presented to the Iowa City Genealogical Society. She's going to be here in May to do that presentation. See you next month.